Okay, continuing on with the group problems, the next question, number 4, asks about the two reflexes important in terms of the cardiovascular system. The first one is the exercise pressor reflex, sometimes known as the exercise processor reflex. This is a reflex that happens when exercise begins. It actually happens before exercise begins, because when we begin to exercise, we need to partition blood to the areas of need. So we need to take blood from the gut and send it to areas of the working muscle that have a higher metabolic demand. And this is accomplished by the exercise pressor reflex. So it's initiated through a feed-forward process. So it happens before even exercise happens. And this is where the body is anticipating a change in blood pressure prior to the exercise. So what happens is we increase stroke volume, we increase heart rate, we increase TPR. We get this global vasoconstriction from the sympathetic outflow, and that's going to help accommodate the changes in blood flow. What happens is we get this massive sympathetic increase and a parasympathetic withdrawal, which helps to lead to the increase in heart rate. The sympathetic increases the stroke volume, while the TPR goes up as well because of the sympathetic system. And then as we start to exercise, those local chemicals that we talked about in the previous TPR section, right, are released into the working muscles. Those all cause vasodilation, right? So we get vasodilation, and that leads to what we call active hyperemia. Remember, active hyperemia was an increase in blood flow due to metabolic demands. So the vasodilation decreases resistance in the vessels, allows increase in blood flow, and blood is shunted from other tissues due to the increase in resistance, and therefore preferentially going to the muscles. This assures venous return, which is an important component, right? We have blood partitioned areas we need, and the venous return, we said how important that was to keep blood pumping from out of the heart. So that's the first portion of the two reflexes. The second one is the baroreceptor reflex. So the baroreceptor reflex is an extremely important reflex, and that helps to maintain our mean arterial pressure. Remember, since mean arterial pressure is a homeostatically regulated variable, it has a set point, and that set point we can get through negative feedback mechanisms. So if blood pressure goes down too much or increases too much, we can see changes that allow us to appropriately move blood pressure back to where we need it to go. The way that happens are the baroreceptors. So baro refers to pressure, receptors, obviously receptors. They tend to be located in the large thoracic arteries, your coronary sinus, in your carotid sinus, in your kidneys. And they detect changes in pressure and then send this information to the cardiovascular centers. And then that's through the cranial nerve number 9, that's the glossopharyngeal. A couple other nerves that do it, but the major one is the glossopharyngeal. And then in your brain, the medulla is the integrating center. It takes this information and other types of information relating to pressure and flow. And then if a change is needed, the medulla signals the ANS to adjust the blood pressure. And so the major ANS nerves would be the vagus nerve, cranial nerve number 10, right? That's the major parasympathetic nerve. Your cardioacceleratory nerve, remember that's the nerve, it's actually a couple of them that go to the heart from the sympathetic side. There's other sympathetic nerves that go to other places besides the heart, blood vessels, kidneys, and adrenal glands. So the way this works, right, is the question asks, okay, if you had a stimulus, okay, so here's my stimulus of 10 millimeters of mercury increase in blood pressure. So our set point, let's say, was 90 for mean arterial pressure, and we went up to 100. Okay, so that's the stimulus, an increase in blood pressure. So blood pressure went up, all right? what's our response? Our response would be to decrease blood pressure, right? So to decrease blood pressure back to our homeostatically set point, right? So our homeostatic set point. So everything on the right side, notice how the arrows go down. So we want to do that. So my stimulus goes up, my response would be to decrease blood pressure back down. Okay, so to decrease. So I like to do the sympathetic first and look at that because that's directly proportional. So if I want my response to decrease blood pressure, then my sympathetic system is going to decrease to do that. So they're directly proportional. Right? The other thing that's nice about the sympathetic is it goes everywhere that we're going to talk about. So there are 4 areas that we're going to be concerned about in terms of the effectors for the baroreceptor reflex. We're going to worry about the heart, we're going to worry about the blood vessels, we're going to worry about the kidneys, we're going to worry about the adrenal glands. And the reason why these are our 4 effectors is the heart will help control heart rate and the strength of contraction primarily, which is stroke volume, right? So those have a huge influence on mean arterial pressure. For vessels, it's the TPR that matters. So that's going to have a big influence on that. For your kidneys, it's going to be the blood volume changes, right? So volume is the long-term regulator of mean arterial pressure, right? And so that's going to help affect how much blood volume we have. And then the adrenal glands, which release epinephrine from those chromaffin cells, which are going to help support what we do on the other areas. So for the output, right, I would draw a heart, a blood vessel, the kidney, and the adrenal gland. And using those, right, we can figure out what's going to happen. So if I want to decrease mean arterial pressure in my response, I put a decrease down here, sympathetic. And if I decrease sympathetic to the heart, that's going to decrease heart rate. We're going to decrease strength of contraction. We're also going to decrease conduction velocity. And all of those are going to help decrease both heart rate and stroke volume. So heart rate and stroke volume go down. For blood vessels, right, I have a decreased sympathetic stimulation. So I'm going to get dilation of the blood vessels. Since the blood vessels get wider, there's less resistance. So that's going to decrease TPR. Right, so that's going to decrease TPR. I'm going to write decreased stroke volume up here for strength of contraction. I already have decreased heart rate here. For the kidneys, we're going to decrease blood volume. You're going to pee more. Since I have less volume, remember volume and pressure are directly related. If I increase blood volume, I increase blood pressure. So if I decrease blood volume, if I have more urination, then I'm going to have a less volume, I'm going to have less pressure. So that's going to help. And then I'm also going to decrease the release of epinephrine from the adrenal gland. Remember, that's the sympathomimetic hormone, or one of them, that will support what the sympathetic system's doing. If I have less epinephrine, I'm going to decrease the stroke volume, I'm going to decrease the heart rate, I'm going to decrease the TPR. Okay? So that's the one set. Now, we can't forget about the parasympathetic system. Remember, the parasympathetic system basically go to the atria, right? And if the sympathetic's going down, then the parasympathetic's going to go up, right? So the increase, notice over here, the arrow, the increase in the parasympathetic. And the only place we have the blue parasympathetic line to is the heart, because there's no parasympathetic stimulation that goes to vessels or kidneys or the adrenal, so it's sympathetic only. And in terms of the heart, if I increase the parasympathetic, I'm going to decrease heart rate, decrease strength, decrease conduction. So that's also going to decrease stroke volume, heart rate, things like that. It has no direct effect on the blood vessels or the kidneys or the adrenals, but it does affect the heart rate. So all of this together will have the net result into decreasing mean arterial pressure, which is exactly what we want to do if our stimulus is an increase in it, right? Our response should be a decrease from the homeostatic point. I'm not going to look at the paragraphs below this because it basically says what I did as a summary. Okay, I just lied. I probably should go over really quick. So again, this explains what I talked about. If there's an increase in mean arterial pressure, say of 10 millimeters of mercury, the baroreceptors would signal the cardiovascular centers in the medulla via cranial nerve number 9. The homeostatic response would be to lower blood pressure back to normal. So we would stimulate the parasympathetic and inhibit the sympathetic, right? The parasympathetic tone of the heart would cause a negative inotropic, that's the strength, chronotropic, that's the heart rate, and dromotropic effect, right? We talked about that in the ANS system. That would lower mean arterial pressure because the heart rate and stroke volume especially would go down. The decreased sympathetic tone of the heart wouldn't have much of an effect because it would probably be rest, but could be any time. So that would have a negative ino and chrono and dromotropic effect as well. We would have decreased sympathetic tone of the blood vessels. Remember, there's no parasympathetic fibers that go to the blood vessels, so because of Postiel's law, we'd have an increase in diameter that would decrease TPR. For the sympathetic tone of the kidneys, that would be decreased. Remember, there's no parasympathetic to the kidneys. That results in an increase in urine output. Since I have less blood volume, my blood pressure will go down. In addition, we'll have decreased sympathetic tone to the adrenals. It's the sympathetic stimulation of the adrenal glands that releases epinephrine into the blood, right? We also call that adrenaline. Therefore, we'll have a decrease in resistance and other things, as it would decrease heart rate and stroke volume as well, because we have a decrease in that sympathomimetic. So there's a sympathomimetic. So, the summary is the baroreceptor helps to control blood pressure short term with minute-to-minute changes. The long-term regulation of blood pressure is accomplished primarily through kidneys and other mechanisms that help control blood volume. Right? And again, the greater the volume, the greater the pressure. So now we'll move on. This time, I promise. Okay, for question number 5, it involves some of the math that we're responsible for on the exam, so I think it's a good one to kind of pay attention to. My suggestion would be to, at the very least, learn this one and make sure you understand how to do this for the lecture exam. But it's not entirely as complicated as it looks initially. I also included the question in this because I think the question is important in terms of understanding what's going on. So what we've asked for is, we said, okay, how can it be a person that had a heart attack, which is a lack of oxygen to the heart, can have more than an adequate supply of oxygenated blood to their left ventricle, right? So if you look at the left ventricle, it's got plenty of blood. And the answer to that, is pretty simple anatomically. The heart gets all of its blood from the coronary blood vessels, right? So the coronary circulation supplies the oxygen. In humans, we don't extract any of the oxygen from our chambers of the heart. So it doesn't matter if there's oxygen in the chamber. It matters whether or not it's in the actual blood vessels of the heart where we can extract it from it. And if you had a heart attack, that means your blood vessel probably was occluded, meaning there wasn't good blood flow, and then we had that area of the heart begin to die because of that. So then the question further asks, if a person does not die from the heart and enters the hospital, right, their cardiac function would be severely affected, possibly, and it's giving you some numbers of this person. So it gives you an ejection fraction of 33%. It gives you a stroke volume of 25 mils per beat, and it gives you a heart rate of 40 beats per minute. So based on these given numbers, ejection fraction of 33%, stroke volume at 25 mils per beat, and the heart rate is 40 beats per minute, answer the following questions. Okay? So it wants you to figure out what stroke volume, ejection fraction, heart rate, cardiac output, end diastolic volume, and end systolic volume are. And you can do that from the numbers given. And then it asks you for the abnormal changes in cardiac function, and it asks you what the scientific name of the abnormal heart rate is. So we can do the abnormal heart rate right now, right? Since the heart rate's below 40, right, it's abnormal, right? Normal heart rate range is 60 to 100. If it's above 100, it's tachycardia, but since we're below 60, this is bradycardia, right? So bradycardia would be the appropriate name. We'll talk about the abnormal changes in cardiac function once we kind of see them with real numbers and stuff. So there are some equations that you should know. This is lecture. So these equations, among others, you should know for the lecture exam, right? You should have them in your head, ready to go. One of the big ones is mean arterial pressure equals stroke volume times heart rate times TPR. Up here, we're asked for cardiac output, right? But we're not asked for other things, but we are asked for cardiac output. Cardiac output, which is often known as Q, is stroke volume times heart rate, right? Stroke volume, that equation is the difference between end diastolic volume, what's in the heart before ejection, minus end systolic volume, what's in the heart after ejection. So the difference between what you started with and what you ended with was the amount you put out. That's the stroke volume. Your ejection fraction is stroke volume times end diastolic volume, right? These should be in parentheses probably to make it easier, times 100, okay? So that's to get the percent, right? So stroke volume is divided by end diastolic volume. So you want to know how much you put out, the stroke volume, over how much you had available, the end diastolic volume, okay? So those equations and others as well, you should have in your head for this, okay? So when you start studying for the exam, you should start looking at these equations, okay? So these equations will be helpful a little later on, all right? So the problem asks you to say, okay, given all this information, right, what do you need to know, okay? So the variables we are asked for is stroke volume, ejection fraction, heart rate, cardiac output, end diastolic volume, and systolic volume. And the variables we were given in the problem, right, are stroke volume, heart rate, and ejection fraction, right? So I can literally just fill those in right now. So I made a little table here, okay? And I did two things. I made a table to put in the numbers that we need, and then also remember the question asked to compare it to normal. So I made a column for the normal values as well, okay? So stroke volume's easy because that was given. So stroke volume was given right here, right? So that's given, so we've got that. The ejection fraction was 33%. Oh, that was given here. We know that. And the heart rate, that was given here, right? So there's our bradycardia. So we already have 3 of the variables done, so we can use these 3 to figure out the other things. Well, nice thing for us right now is we have cardiac output. So if we go back to the top, and we have the formula right here as well, so we don't need to, is cardiac output is stroke volume times heart rate. So Q, the cardiac output, is stroke volume times heart rate. So all we need to do is take the 2 given numbers, stroke volume and heart rate, 25 and 40. So 25 mils per beat times 40 beats per minute, right? The beats cancel out, and we get 1,000 mils per minute. So I put 1,000 mils per minute here. Remember, units matter most of the time. Okay, so we can figure out that. Now we get to, probably the one that's a little more difficult, and that's figuring out end diastolic volume. Okay, to figure out end diastolic volume, okay, all we have to do is reverse the ejection fraction, right? So the ejection fraction equation, let's see, it's peeking through right at the top. So the ejection fraction equation is stroke volume divided by end diastolic volume times 100, okay? So if we rearrange that, right, and replace end diastolic volume, right, with ejection fraction, right, we get this equation right here. Stroke volume divided by ejection fraction divided by 100, right, gives me my end diastolic volume. So we know stroke volume, that's 25 mils per beat, divided by ejection fraction, that's 33%, so that's 33, divided by 100, right? So that's that equation there, just replacing these 2 variables, ejection fraction and end diastolic volume. Then you punch that in the calculator, you get 25 divided by 0.33, which is 75. So you get an end diastolic volume of 75. To get to end systolic volume, right? That's the amount of blood left in the heart after ejection. We know what we started with, end diastolic volume, and we know what we ended with, stroke volume. So all we have to do is take the difference between stroke volume, right, and end diastolic volume. So end systolic volume is end diastolic volume minus stroke volume. So we know those numbers, 75 and 25. So we get 50 mils. It's actually technically 50 mils per beat, but 50 mils. So we can figure those out. All right. So again, knowing these equations and their derivatives will allow you to calculate this information here, given the numbers that we had for the cardiac patient. Now, you're also supposed to compare it to normal. When we look at normal, we say, okay, the stroke volume, should be 70 mils per beat, right? The stroke volume for this person is only 25, so that's down. So that's decreased. The ejection fraction should be up at 70%, so it's 33, that's decreased. The heart rate should be about 70 beats per minute, right? It's at 40 because it's the bradycardia, so it's decreased there. Because heart rate and stroke volume have gone down, it makes sense that cardiac output, which should be about 5 or 5.5 liters per minute, which is about 5,000 mils per minute, right? It's down for our patient at 1,000 we calculated, so it's down, right? But here's where it's a little different, okay? Our end diastolic volume is up, and it could be up or down depending on the patient. Normally it's about 100. Oops, sorry. Look at the wrong one. The end diastolic volume is down. Okay. And that's because cardiac output's low, so venous return is low. So the blood isn't filling as much in the heart as it could. Even though the heart rate's slower and you have more filling time, you don't have adequate venous return, so that goes down. The one I was talking about that went up is this one here for end systolic volume. And the end systolic volume is up. So I'm going to change color. So, okay, this one went up. So it's the only one that went up. So normal, it should be 30, right? And in our patient, it's 50. And remember that end systolic volume is that highly regulated variable that has to do with the Starling's law of the heart, that the end diastolic volume, if we increase the end diastolic volume, we increase the amount of blood we can pump out, and it's the pump what you get. The reason why we don't pump what we get this time is this person just had a heart attack. And what the heart attack is going to do, it's going to damage the muscular tissue. And with the damaged muscular tissue, we're not going to be able to pump out as much blood because it's damaged. So you can't say all of them, right, went down. We can say that most of them went down, but end systolic volume went up because my heart is damaged and it's not working as well as a pump, and which is the answer to that last question that we saved for the end. So that is what happens with a cardiac patient and some of the ways to look at these numbers. And you should practice this and kind of understand how it works. Okay, on to the sixth question. It's a warm sunny day and Mike's outdoor wedding. He suddenly faints. Like you see on those videos, right? The person just fainted in the middle of the wedding. Mike has been standing very still, his knees locked, he's listening attentively. It's a warm, sunny day, and let's kind of see what happened to Mike. To help us kind of figure some stuff out, a doctor took Mike's blood pressure, who was there, and noted his blood pressure was 90/60, so we should explain that. We should try to calculate his mean arterial pressure and his pulse pressure from these figures and kind of talk about that a little bit. In addition, given that Mike has a heart rate of 120 beats per minute, so we can talk about why it's high. His stroke volume is 58, a little over 58. We can talk about why that's low, right? And his TPR as well. And so he has a low TPR. We can talk about that. So we can calculate his arterial blood pressure, and we can look at the two methods for calculating, and we'll actually look at three methods, why and why not. So how did all this contribute to what happened to Mike? So let's kind of scroll down a little more. All right, so what we're looking at, right, is when Mike is standing there with his leg locked, that'll diminish his venous return. The reason why is, remember, we talked about the muscle pump, and because of gravity, blood is going to pool in his legs, okay? And so if he's standing there with his legs locked, he's not going to be able to return blood back to his heart, so he's not making use of that muscle pump. Also, because it's hot outside, we have a high temperature. And what's going to happen with the high temperature is some of his blood is going to go selectively to his skin. You send blood preferentially to the surface of your body to cool it, right? So he has less blood centrally. So his core blood, and especially going to his brain, is less because of, two major reasons. One, a lot of it is going to the skin because it's vasodilated, and some of it is being pooled in his leg because it's not returning because of gravity. Okay? That means that since there's less venous return, we have less cardiac output. So there's again that association between venous return and cardiac output. Since he can no longer pump sufficient oxygen to his brain, right, he faints. His blood pressure was measured at 90/60, we saw, right, which gives us a pulse pressure of 30. You just take the difference between systolic and diastolic. So 90 minus 60 is 30. So his pulse pressure is 30. This lower pulse pressure, right, indicates a low stroke volume, right? Remember, stroke volume and pulse pressure are related, okay? And it's a good estimator, pulse pressure or stroke volume. So it's lower than normal. Normal would be 40. It's 30, so it's gone down. His mean arterial pressure is 70. We can figure that out a couple ways. I think the easiest way is to take 1/3 of the pulse pressure. That's this one. 1/3 of the pulse pressure plus diastolic. So 1/3 of 30 is easy. It's 10. I can do that in my head. And diastolic I don't have to do anything to. It's given as 60 right here, right? So here's my 60. So 10 plus 60 is 70, right? The other way to do it is 1/3 systolic. And in this case, it's not too hard. Systolic is 90, so 1/3 of that is 30. Plus 2/3 diastolic. Diastolic is 60, so 2/3 of 60 is 40. So I get 30 plus 40 is 70 anyway. So no matter how I do it, right, I end up with, the same mean arterial pressure, 70, which I should. Okay, so his mean arterial pressure is down because of the pooling of his blood in his legs and the blood going to the skin. He has a reduced stroke volume. We saw that it was given as 58.34, right? And normal is 70, so it's a decrease of 20%. It doesn't look like it's gone down that much, but it's decreased to 20%. Think about this, physiological variables change day-to-day variation is probably less than 5% for most of the variables, and usually for many of them significantly less than that. So any variable change more than 5% usually is something indication of something significant. Or look at it another way. For those of you that have jobs, think, oh, there's not that much difference between 70 and 58.34. What would happen if you went into work tomorrow and they said, oh, we're going to decrease your pay 20%, right? That would really suck. So, it is a pretty significant decrease. So the decrease in stroke volume is the decrease in venous return. Remember, we try to increase venous return to do that. And in fact, your body fainting is a way to increase venous return because now you're flat, right? Instead of standing in the field of gravity, you're flat. And that's going to increase venous return, which is going to help increase stroke volume so your blood can get to your brain. Okay. So in order to try to compensate for that decrease in stroke volume, right, our cardiac output's going to go down. So Mike becomes tachycardia, right? So he gets tachycardia, his heart rate goes up. So Mike increases his heart rate to try to compensate for the decrease in stroke volume. Remember, cardiac output, right? Cardiac output is equal to heart rate times stroke volume. So if heart rate went down and we didn't want cardiac output to go down, we have to increase stroke volume. So, and if you look, his increase in heart rate is actually more than compensating for that. his average stroke volume is about 5,000 mLs per minute, and he's right now, if you do the math, is about 7,000. Okay. In addition, the heat contributes to Mike's higher than normal heart rate as well. Remember, we said as you increase heat, as increased temperature, heart rate goes up as well. So that contributes to it. All right. So we can also calculate mean arterial pressure given those numbers. So we know Mike's heart rate is given as 120 beats per minute. We know his stroke volume, 58.34. We know his total peripheral resistance, 0.010 millimeters of mercury times minute over mLs. So we can calculate those variables as well, right? So we can put those into the equation. And we look down here. And we see put it right here. We put in this equation, mean arterial pressure, heart rate, 120 beats per minute, times 58.34 mils per beat, times 0.010 millimeters of mercury times minute times ml, equals 70 millimeters of mercury. If you remember back at the top, based on his blood pressure, we also calculated the same mean arterial pressure. No matter how you calculate it, it should be the same, right? One note about units here, right? Since mean arterial pressure units is in millimeters of mercury, right? When we do the cardiac output calculation, the beats cancel out, right? So we're left with minutes and mLs, right? So to get to millimeters of mercury for our units of pressure, right, then what we do is we have to have those PRU units, those peripheral resistance units, so that it cancels. So since we got minutes on the bottom here, we got to cancel with minutes on the top for the TPR. Since we have mLs on top here, we don't need it. We have to cancel with the mLs on the bottom here. And all we're left with is the millimeters of mercury unit. So that's why you have these weird units for pressure. So basically, you know, Frank's not getting enough blood back to his heart because of the decrease in venous return, because of the pooling of the blood in the legs, because he's got them locked, because of the heat, he's sending blood to his skin and trying to vasodilate it and release heat to the exterior of the body to help control blood pressure. His body temperature is probably up, so his heart rate's up. Because of the decrease in venous return, you have a decrease in stroke volume, so his heart rate's going up to try to compensate for that, to keep cardiac output high enough, and it actually has to go up even more than normal because of the pooling of blood in the skin and the pooling of blood in the legs. So all of that contributes to him fainting. One interesting side story, years ago, I gave this question to one of the groups, and one of the students kind of basically said, what did he say? He said, why you do me like that, James? I said, what? And he said, why you do me like that? Well, I didn't know what he was talking about. Turns out name was Mike. I knew that. But I did not know he had gotten married over the summer and fainted. He got married at a golf course on the 18th hole for some reason and actually fainted during the ceremony. So he thought I knew and wrote it for him, but I had always had Mike as that person. So that's kind of weird. So we'll look at one more that's really not part of the group problems technically, but we'll just go over it really quick. So question 7 is the bonus question. And if you know, all of the previous questions were on the cardiovascular lecture notes. And this question is just to remind you, hey, there's also blood, right? So don't forget about the blood. And basically this question is pretty general. It says identify the major constituent of blood and explain the functional importance of each, right? So basically the function of the blood in general, transport, right? Blood takes oxygen to cells, nutrients, hormones, water to cells, and removes CO2 and other waste from cells. In terms of protection, it protects from a number of different things. First of all, it's got the immune system, right, which helps protect us from disease, microbes, things like that. The other protective mechanism is that blood clots, so we don't lose all of our blood. So it's a self-protective mechanism. It also helps to regulate temperature. We kind of saw that on the previous question where Mike fainted on the hot golf course, right? And blood goes to the surface of the body when hot, right? Or when we exercise, we turn red, right? That showing an increase in blood flow to the area, the active hyperemia to release heat. When it's cold out, blood goes to the central area of the body to keep the vital organs warm, so we send less blood to the periphery in those cases. So temperature regulation is important as well. When we look at blood, we can break it down a couple different ways. We can look at whole blood. Whole blood is composed of cells. Those are the formed elements, the erythrocytes, leukocytes, and thrombocytes. And plasma, right? That's the liquid in the cells, where the cells are suspended. And within the plasma, there are solutes, things dissolved in the plasma. As we said before, plasma is mostly water. A good estimate of total blood volume is about 5.5 liters. Okay, although really the better estimate is of a mammal at least, is 8% of the total body weight in kgs. That's probably a better way to look at it. Your plasma makes up about 60% of your whole blood. And we have about 3 liters of plasma, since our blood is 5 liters roughly, and 60% of that would be 3 liters in terms of that. So, from a standpoint of standardized tests, like if you're taking some sort of nursing exam or some other exams, and they ask you what's the total blood volume in a human on average, the answer is probably going to be 5.5 liters. But as I said before, 8% is a much better way to estimate that, because we're all different sizes. And remember that 5.5 liters is for that magical medical male model, right? Who's, college age and 70 kilograms and things like that. And there's very few of us that actually fit that description, right? So, as an example, just to kind of look how it works, if someone weighed 100 kgs and you took 8% of that, they would have 8 kgs of blood. And again, we're just going to assume that blood is same as water in terms of its weight. So 8 kgs of blood is about 8 liters of blood. So this person would have 8 liters of blood. That would be their total blood volume. Right? And the bigger you are, obviously, the more blood you would have. The liquid portion of whole blood is we call plasma, right? It's mostly water, like over 90%, with some dissolved particles. Most of the dissolved particles are proteins, but there's other things we'll see in a minute. If we remove the clotting factors from blood, it becomes serum. The cellular portion of blood is known as the formed elements. There are the erythrocytes, those are the red blood cells. Their primary function is to carry oxygen, although this also carries CO2 and serve as a buffer. Leukocytes, those are the white blood cells. Their primary function is immunity and all sorts of different types of immunity, keep us free from disease. And the last one are the thrombocytes. Those are our platelets, and their primary function is clotting. So let's kind of break down the plasma a little more. Plasma is a liquid portion of blood. It makes up, like I said, about 40 to 50% of blood in most people. The difference between how much plasma you have really depends on how much your red cell hematocrit is. So your packed red cell volume is called the hematocrit. We'll see that in a couple minutes, but your plasma is the difference between that and 100%. Plasma is about 90% water with dissolved particles. A lot of the dissolved particles, about 80% of those are proteins. There are actually a couple different proteins that are important in terms of the plasma. The albumins, which make up more than half your proteins, right, are the osmotic regulators of the body to a large extent, and also they help to add viscosity to the blood. You have gamma globulins. Those are proteins for the immune system. They are the antibodies we make. These are the most diverse set of proteins because we have a specific antibody for every antigen. And we come in contact with literally potentially millions of antigens over our life. So we have millions of different antibodies. Fibrinogen, that's important for blood clotting, right? The eosin tells me it's inactive. When it becomes activated, it becomes fibrin and makes the network to catch the platelet plug. And also not listed here for some reason would be the various protein carriers. Remember, if something is non-polar, like a steroid hormone or cholesterol, it needs a protein carrier in the blood in order to dissolve in the blood and be carried without causing the blood to kind of get all thick and not flow. Other things in plasma are your ions. Those are charged particles such as sodium, potassium, calcium, chloride. You also see gases, oxygen, CO2, nitrogen are all dissolved in blood. And nutrients like glucose and amino acids can be found in blood as well. So those are the constituents of plasma. Your red blood cells, right, are your major constituents in terms of the cellular component. Remember, those are called formed elements collectively. So all of your cellular components, your red blood cells, your white blood cells, and your platelets are known as the formed elements. For the red blood cells, we could have a total red blood cell count. That's approximately 5 million per cubic millimeter of blood. That's also known as a microliter of blood. Okay, so it's a very, very small amount. They're biconcave discs because that increases the surface area and allows greater diffusion of oxygen. They're very flexible to bend through capillaries. As a matter of fact, our liver and our spleen, as blood goes through it, there are cells that line the sinusoids. Those are kind of the tortuous capillaries of the liver and spleen called Kupffer cells, and they remove the red blood cells from circulation if they're not flexible. And like us, as they get older, the red blood cells lose flexibility, and so they get recycled. Red blood cells are anucleate. They have no nucleus. What we've done is we've scooped out almost everything we can out of a red blood cell and replace it with hemoglobin. The more hemoglobin we have in the red blood cell, the more oxygen it could carry. Hemoglobin is a protein. It has about 270 million hemoglobin molecules in each red cell. Each hemoglobin molecule can carry 4 oxygen molecules because there's 4 heme groups and it's carried on a heme group. When oxygen is carried on a hemoglobin, it's called oxyhemoglobin. When there's no oxygen on it, it's called deoxyhemoglobin. It's the hemoglobin that gives blood its red color because of the iron, and when oxygen binds to it, you have an oxidation reaction which causes it to turn red. All right. When there's no oxygen on the hemoglobin, the deoxyhemoglobin turns blue. We call that cyanosis. Cyan is another name for blue, and you'll see that sometimes, medically speaking. Hemoglobin carries other things, though. As an example, it's responsible for carrying some of our CO2. It's actually about 23% or so. But if it's carried as CO2, it's not carried on the heme group. It's actually carried on the amino acid portion. So it's called carb-aminohemoglobin. Carb referring to CO2. That's often the scientific words we use for that. Amino because it's carried on the amino acid. Hemoglobin because it's hemoglobin. So it's that. Or we can also carry carbon monoxide. That's called carboxyhemoglobin, right? That's about 250 times more affinity for oxygen. So carbon monoxide poisoning can be very serious because the carbon monoxide will preferentially bind to heme, and that serves as a competitive inhibitor of oxygen. So if you have too much carboxyhemoglobin, you'll die because you can't get enough oxygen. You also have methemoglobin, which the heme is not in the right state, and oxygen can't bind to it. Hemoglobin also serves as a buffer for hydrogen in red blood cells, so it helps to maintain pH. For red blood cells, some other values you should know, right? We said that the number to remember magically is about 14 grams per deciliter for hemoglobin in the blood. Men are like 14 to 20, women are 12 to 15, and infants are 16 to 22. But the magic number, everybody, for infants maybe not, but for everybody else, 14 is a good number, so it's kind of like we remember 14. The hematocrit, your packed red cell volume, Again, men tend to be about, 40 to 50, and women tend to be about 38 to 46, but the magic number for that is 42. And remember, if we use 14 and 42, there's that one-third hematocrit is your hemoglobin, and that works pretty well for most people. For white blood cells or leukocytes, they're about 5,000 to 10,000. Okay. If you have a decrease in your white cells, that's immunocompromised. That would be when your white cells are below 5,000, right? So you're said to be immunocompromised. That would be leukopenia. If you have an increased, it often comes from fighting an infection. So you can do that as well. That would be leukocytosis. We can divide them up into granulocytes and agranulocytes. And actually, we're going to have to start this on a new video probably, but we'll see. We'll keep on going and see if it uploads. So for the granulocytes, they have granules in the cytoplasm. We have neutrophils, which make up 60 to 70% of your blood, eosinophils, which make up 2 to 4%, and basophils, which make up 0 to 1%. We also have the agranulocytes that have no granules in the cytoplasm, and that makes up about 20 to 25% for lymphocytes and about 4 to 8% for monocytes. And we can use the never let monkey eat bananas moniker to remember from most numerous, never, N, neutrophils, L, let, lymphocytes, M, monkeys, monocytes, E, eat, eosinophils, B, bananas, right, basophils. Let's take a look at the individual white blood cells and kind of see what you have to remember about each one. So neutrophils make up about 60-70% of the white blood cells. They are called neutrophils because of their neutral staining granules. They are identified by a multi-lobe nucleus that's irregular in shape most of the time. They are capable of diapedesis. That means they can leave the bloodstream through a capillary and go to the tissue to help fight an infection. As a matter of fact, all of the white blood cells are capable of diapedesis except for basophils. Their main job is phagocytic. They destroy other cells. Their high count helps in terms of bacterial infection, and they have a very short lifespan. In some cases, they last only seconds. The eosinophils are about 2 to 4% of the white blood cells. The eosin means reddish staining granules, so they have these red granules. They often have a bilobed nucleus. They are capable of diapedesis. They are great at fighting parasitic infections, so they're good at doing that. And they make antihistamine to help protect against hypersensitivity. Hypersensitivity would be allergy. So as an example, you might take Benadryl, right, that had an antihistamine if you had an allergic reaction. Basophils are your next. They are less than 1% of your white cells. They are dark staining purple granules. They has an S-shaped nucleus that you often can't see because it's occluded. They increase in allergic reactions. They release chemicals that help the inflammatory response. They are similar to mast cells that are found in the tissue. They cause vasodilation, which increases capillary blood flow by releasing histamine. They also release serotonin, which increases capillary permeability. And they also release heparin, which is an anticoagulant. We've used that in lab to keep the tubes of blood from clotting up. Monocytes are the next one. There are 3 to 8% of the white blood cells. They are the agranulocyte, right? So they're in a granulocyte. They have no granules. They're the largest white cell, and they typically have a pale stained bean-shaped nucleus. Again, they can do diapedesis. They can also evolve into other forms of blood. As an example, other forms when they leave the bloodstream. When it's in the bloodstream, it's called a monocyte. When it leaves, we change the name. So as an example, if it leaves the bloodstream and enters the tissue, we call it a macrophage. And then sometimes we even give it a more specific name. So the bone monocytes, right, that left the bloodstream become macrophages, and now we call them osteoclasts in bone, and they're responsible for the absorption of bone, as an example. They are much stronger and more effective white blood cell than the neutrophils. They are slower acting, though. But the neutrophils die really quickly because they kill like one thing by phagocytosis. This can do many, many. High monocyte count often means, because they're slow, you have a chronic infection. In addition, they can be made in the lymph or bone marrow. Remember, so lymph means lymphoid production, bone marrow means myeloid production. All of the white cells can be made in myeloid area, right? So they have myeloid production. They can be made in the red bone marrow. Matter of fact, all of the formed elements can. And only the monocyte and lymphocyte can have lymphoid. For the lymphocytes, they make about 20 to 25% of your white blood cells. They're found primarily in lymph tissue, but also the red bone marrow. There's a few in blood, which only about 2 to 10% of them. You see, there's no granules. They have a large stained nucleus, but they're the smallest white blood cell. Tends to be a little darker staining, although there's different types. There's 2 types with subdivisions of each type. The T cells are thymus derived because they mature in the thymus. The B cells are bone marrow derived because they mature in the bone marrow. They have many functions. We're not going to worry about what function is related to what lymphocyte, but they make antibodies. They're responsible for immunomemory. They can cause allergies, and they're responsible for organ rejection if somebody has like a transplant. The last formed element, the thrombocytes, also known as platelets, they are small fragments of megakaryocytes. They're made in the red bone marrow and then basically split into many, many platelets. They're an active tissue. They have no nucleus, but they have endoplasmic reticulum, so they make chemicals. They're very important for clotting. On a slide, they look like purple dots because they're much smaller than the red blood cells and white blood cells. They have a short lifespan, on average about a week, and there's 200 to 400,000 mLs in blood. While not on here, also do not forget about the hematopoiesis section as well. So that concludes this, and hopefully this one uploads and it's not too big a file.